In yesterday's video, we looked at more of the function of DNA, and today it is all going to be about the structure. And it's also worth noting that you guys already took notes on this, so as I'm going through this lecture, it would probably be a good idea to take a look at page 4, where you took the notes on chapter 12.1 and 12.2, and then just add in any notes that you think are relevant or important when you go and color the image on page 5. Hopefully that helps elucidate some of this information as well, but there are no actual questions in your packet related to this lecture. To really understand the structure of DNA, we need to take a little bit of a historical backtracking and look at some of the people that contributed to understanding the structure of DNA. The first of which is going to be my man's Edwin Chargaff. And so he was a chemist, and he was analyzing different samples of DNA from chickens, from, I don't know, other stuff. And he realized something really interesting was happening. So scientists didn't know what the structure of DNA was, but they did know that it was composed of nitrogenous bases, so adenine, thymine, cytosine, and guanine, as well as phosphate groups and sugars. And what Chargaff realized is that in every sample that he analyzed, the amount of adenine and the amount of thymine was always the same, and the amount of cytosine and the amount of guanine was always the same. And so that really struck him as something interesting. But he couldn't quite figure out why that is. So we're going to have to come back to figure out what was going on there. Rosalind Franklin, as you saw in the video, was one of the two scientists that worked a lot with X-ray crystallography, which is basically taking different substances. In this case, she was working with DNA, and putting it into a crystalline form, and then shooting it with X-rays and seeing where it diffracted. So this X-ray diffraction pattern, this picture up here, that X shape is characteristic of things that have a helical shape. And again, she didn't put too much thought into it. She kind of said, okay, DNA is in a helical shape, but nobody really cares about DNA anyway because proteins are the genetic information. Yay. So she kind of brushed it off, just knowing that it meant something, but not really looking too much into it. And as you saw in the video, James Watson and Francis Crick, these guys are the ones that really take the information from everybody else and put the puzzle pieces together. They were able to really reason and synthesize, look at that, 2 of your thinking skills, these different pieces of information to determine what it really meant. So by using the fact that A and T always appear in equal amounts, and the fact that C and G always appear in equal amounts, they were able to figure out that, well, maybe it was the nitrogenous bases themselves that were pairing together. And looking at Rosalind Franklin's work, they were able to say, okay, this is helical, maybe it's double helical, meaning there are 2 strands that can then bind together. And they figured out the structure and made a 3-dimensional model, and in doing so, published one of the most significant findings in biology, and that did win them the Nobel Prize in 1961, I believe. So when we look at DNA, it is, just to kind of reiterate, it is the code for making proteins. If you don't remember about proteins, let's go over some of the functions of those really fast. So some of the some of the functions of proteins include hormones. So for example, insulin is a protein hormone. So this protein gets secreted to tell the cells when to take up more glucose. Your immune system, anytime You recognize something as an invader so you can phagocytize it. That is going to be done via proteins that are on the cell surface and embedded in the membrane. And speaking of the membrane transport, if we're thinking channel proteins, those are, as it sounds like, proteins. What allows things into and out of cells tend to be proteins. Pumps also are proteins. We can look at structure. The cytoskeleton is largely made out of proteins, and of course muscles, and structure and muscles really go together there. And then enzymes. And what's really important about enzymes, of course, is that these enzymes are what make up most of the other tissues in your body. So even if something's not made of proteins, it was made by a protein. So really everything that makes you comes down to proteins, and the instructions for making those proteins are all about DNA. Okay, so role of DNA is to code for proteins and be able to carry the genetic information, which means that it has to be copied each time one cell splits into two, and that genetic information can then be passed from one organism or from one generation onto the next. And I guess those second two really kind of go together there, but there you go. Okay, if we're taking a look at the structure of DNA, what is going to be important for you to know is, for starters, we have nucleotides that are in a repeating fashion. So to know what a nucleotide is going to be pretty important. So a nucleotide is the monomer of nucleic acids, and they are composed of a phosphate group right here, a 5-carbon sugar. So we've got 1, 2, 3, 4, 5 carbons there, as well as an oxygen, and then some kind of nitrogenous base. In other words, a chemical that has nitrogen in it. Okay, so a phosphate, a 5-carbon sugar, and a nitrogenous base. You should definitely be able to draw this out. So even if you just draw a really simple kind of stick model where it's like circle, pentagon, and then like rectangle to represent the nitrogenous base, that is totally fine. Then understanding how these nucleotides polymerize and join together is going to be important as well. So what we end up with is that a covalent bond is going to form between the sugar of one and the phosphate of the next. The sugar of one and the phosphate of the next. So all of these being shown right now are covalent bonds. And so we've got this kind of sugar phosphate, sugar phosphate, sugar phosphate backbone is what we're going to call this. So anytime you hear me refer to the backbone of DNA, that's really what I'm talking about, is this repeating sugar, phosphate, sugar, phosphate, sugar, phosphate. And then in kind of pointing off to the side are where each of your nitrogenous bases are. So that's going to be these guys pointing off into off to the side. And in the case of DNA is a double-stranded molecule. That is, it is made out of 2 strands of DNA. So we have, let's go ahead and take a second in your mind here and identify how many nucleotides are there, how many different pieces of monomers there are. And then I'll go ahead and outline them. So we've got about 8 nucleotides being shown here, and what's important to see is we've got the phosphate group, the 5-carbon sugar, and the nitrogenous base. So this right here is going to make up one nucleotide. We've got that sugar phosphate backbone, and if we take a look at the other strand, that's running in the opposite direction. So whereas this guy, I kind of almost imagine it as the sugar pointing up, because the O is on this side, and that's where the phosphate is as well. The other side has the sugar pointing down and the phosphate down here. And so for that reason, we're going to describe DNA as running anti-parallel. That is, the 2 strands are running in opposite directions. And the 2 strands are each held together within the strands with covalent bonds. So all of these solid lines right here represent covalent bonds, and each of the nucleotides are held together with covalent bonds, and these are really, really strong. You're not going to break these as easily. But remember what a hydrogen bond is, and it's kind of worth noting. We saw hydrogen bonding when we were looking at water, and the reason is because oxygen has a partial negative charge, and that's going to pull the electrons kind of towards it, right? So that's what's making the oxygen negative. And as a consequence of the oxygen being slightly negative, the hydrogens end up a little bit positive. So a hydrogen bond is that weak attraction that is forming between the between a slight negative and a slight positive charge on different molecules. Okay, and again, they're really weak. They're really easy to break. But in this case, because of all the nitrogen, and because nitrogen is slightly electronegative, we're going to end up with parts that are partially positive and partially negative, and those parts are going to be complementary or, opposite to each other on A and T, and then on C and G. And so for that reason, we can have C and G bind because the hydrogen bonds fit just perfect like a puzzle piece between them. So again, these right here are weak bonds. So in between each of the two strands of DNA, what is holding these two strands together are weak hydrogen bonds. Okay, at this point, there's a video up on Schoology. It would be wise of you to go ahead and look at that. It does a really nice job of showing what the structure of DNA looks like kind of from a diagram perspective as well as in a three-dimensional model type perspective. Once you watch that video, go ahead and come back to this one, and I'll finish up with the lecture for you. Okay. This wasn't really in the reading, so I think it would be a wise decision to, down at the bottom of your notes for chapter 12.1, 12.2, just use some of that space to outline this. We're going to be coming back to it later, but I think it's worthwhile to view. Okay, so when we're taking a look at DNA, here are each of the nucleotides, and this again is shown in a diagrammatic view, so it's just a little bit easier to draw out and view what's going on. So each of what I'm drawing squares around right here are different nucleotides. In green, now what I'm drawing squares around, each of these are base pairs. In other words, nitrogenous bases that pair together, right? And then we start getting into that double helix form where it starts spinning. There are 3 billion base pairs in your genome. That is a ton of genetic information and a ton of molecules. And to be able to pack that into every single nucleus of your body, of every cell in your body, it's got to be packed really tight. So what we're looking at here is DNA packaging. How is it that DNA fits to get inside your cells? And so what it's important that you understand here is that this kind of double helical shape is going to start to coil, right? And we're getting this tighter and tighter coiling until eventually the coils even start to coil. And you don't need to know the term there. And so continued coiling is eventually going to lead to these guys that you've probably seen before. A chromosome is one package of DNA, as well as all the proteins that are involved in packaging it. Remember when the discovery of the structure of DNA came about, it was probably put off a lot because of the fact that so many people thought that the proteins that were in the chromosomes were more important in terms of genetics. So chromosomes are made out of proteins and nucleic acids. Had they realized just how important the nucleic acid part would be, they probably would have studied it more and found the structure sooner. Regardless, a chromosome is just a single molecule of DNA with hundreds of thousands or millions or more of base pairs that is super tightly coiled together until finally you end up with this kind of shape here. So that is a chromosome. It's worth mentioning that in a human, you have 23 sets of different chromosomes. Everything kind of down here, let's go ahead and hold off on all of that information until later in the unit. But what I do want you to know is that you have 2 sets of 23 chromosomes, one that you inherit from each parent. Okay. Anyway, hopefully that gives you a little bit of understanding about the structure of DNA. You've seen really good diagrams of it, and hopefully you've colored the double helix that is on page 5 of your packet. Once you've finished this video, go ahead and look at the Journey into DNA. There's a link on Schoology and questions on pages 6 and 7 of your packet, at which point you can then, after you're done with that, go through the questions, and you should be all set to go for this packet. If you've got questions, that's why I'm around. So, all right. Bye-bye.